Cells constantly hydrolyze ATP to provide energy. Describe how ATP is resynthesized in cells. So this question is asking you how you make ATP. And the two molecules we require are adenosine diphosphate, that's ADP, and an inorganic phosphate. And it's important to say inorganic phosphate, not a phosphorus, otherwise you won't get the mark. And that's converted into ATP by an enzyme called ATP synthase. And this happens during photosynthesis and respiration. And these points are actually, all three of them, directly written on the specification. So if you'd had a look at that, you'd know exactly what to say to get the marks here. And if we just look at the mark scheme for that, we'll see those are the 3 points there. And there it says reject phosphorus. Okay, give 2 ways in which the hydrolysis of ATP is used in cells. Now, at first this seems really obvious. We automatically associate ATP with energy, which is correct. ATP releases energy, and this would be our first point, releases energy, but you need to say a specific reaction that it could be used in. My go-to one would be active transport. Now, what a lot of students did is they just gave 2 reactions that energy was used in. But actually, it's 2 ways the hydrolysis of ATP is used in cells. So what else do we get from the hydrolysis of ATP? Well, we also get an inorganic phosphate. So this inorganic phosphate can be used to phosphorylate other compounds. And what this does is makes them more reactive. And an example of this is in second year when glucose is phosphorylated during glycolysis. And if we have a look at the mark scheme again, here we have it. So 1 mark for something to do with energy, 1 mark for the phosphate. And actually, only 3% of students got this second point on top of the first one, which really shows the importance of looking at the specification, in which case you would have seen that you need to know about both energy and the phosphorylation of other molecules. Okay, third part. So figure 2 is a photograph of a mitochondrion taken using a scanning electron microscope. What is the evidence, so what can we see in figure 2, that a scanning electron microscope was used to take this photograph? So first thing I do when I see this question is think about what topic is this testing your knowledge of? It's testing your knowledge of microscopes and how they work. And what do we know about scanning electron microscopes and how they differ from transmission electron microscopes? Well, the main reason that they differ is because they scan electrons across the surface and therefore create a 3D image. And you can see here that this image does look kind of 3D. So that's how we know a scanning electron microscope was used to take this photograph. And here we have the mark scheme. So you can see a 3D image. Name the part of the mitochondrion labelled X in figure 2. So the part labelled X there is these little folds of the inner membrane, and these are just called cristae. That's just something that you have to learn. The actual length of the mitochondrion between points A and B in figure 2 is 4 micrometers. What is the magnification of the mitochondrion in figure 2? Show your working. So the equation we're going to use is I am. And this is image, actual size, magnification. So if we want to work out magnification, we have to do image divided by actual. So first we need to measure the actual size of it in the image, and I've measured that to be 84 millimeters. And the actual size is 4 micrometers. But with all calculation questions, you need to pay attention to the units. Now, there's lots of different ways you can approach doing these units. I find it's easiest to convert everything into meters. This little m here, this means times 10 to the minus 3 meters. And this micro here means 4 times 10 to the minus 6 meters. And I think this is the easiest way to do it. That should give you an answer of 21,000 1,000. If you did it a different way, maybe you got mixed up with the wrong way round, you'd have got a weird number for magnification, and that would tell you that you probably need to try a different strategy, a different calculation, until you get an answer which seems plausible, which I think this one does.